Good day, this is Professor Will, CBMD PhD. Today is Friday, March 26, 2021. It is 6:00 and 8 seconds a.m. Eastern Standard Time. Today's research, muscle cells. So let's hop to it here. Zooming in on muscle cells. Date March 24, 2021. Source, Max Planck Institute of Molecular Physiology. Summary. An international team has produced the first high-resolution 3D image of the sarcomere, the basic contractile unit of skeletal and heart muscle cells, by using electron cryotomography. Electron cryotomography capability of imaging structures directly in frozen muscle cells can translate into future medical treatments for muscle diseases and a better understanding of the aging process. Sarcomeres are small repeating subunits of myofibrils, the long cylinders that bundle together to make the muscle fibers. Inside the sarcomeres, filaments of the proteins myosin and actin interact to generate muscle contraction and relaxation. So far, traditional experimental approaches to investigate the structure and function of muscle tissue were performed on reconstructed protein complexes, or suffered from low resolution. Electron cryotomography instead allows us to obtain detailed and artifact-free 3D images of the frozen muscle. Cryo-ET was for a long time an established yet niche methodology, but recent technical advances in electron cryomicroscopy, cryo-EM, as well as the new development of cryo-focused ion beam, FIB milling, are pushing cryo-ET resolution. Similar to cryo-EM, researchers flash freeze the biological sample at a very low temperature, minus 175 degrees Celsius. Through this process, the sample preserves its hydration and fine structure and remains close to its native state. FIB milling is then applied to shave away extra material and obtain an ideal thickness of around 100 nanometers for the transmission electron microscope, which acquires multiple images as the sample is titled along an axis. Finally, computational methods reconstruct a three-dimensional picture at high resolution. Researchers performed cryo-ET on mouse myofibrils isolated at the King's College and obtained a resolution of 1 nanometer, a millionth of a millimeter, enough to see fine structures within a protein. We can now look at a myofibril with details thought unimaginable only 4 years ago. It is fascinating. The calculated reconstruction of the myofibrils revealed the three-dimensional organization of the sarcomere, including the subregions, MA and I bands, and the Z disc, which unexpectedly forms a more irregular mesh and adopts different conformations. The scientists used a sample with myosin strongly bound to actin, representing a stage of the contracting muscle that is called the rigor state. And indeed, they can visualize for the first time in a native cell how two heads of the same myosin bind to an actin filament. They also discovered that the double head not only interacts with the same actin filament, but is also found split between two actin filaments. This has never been seen before, and shows that proximity to the next actin filament is stronger than cooperative effect between the neighboring heads. This is just the beginning. Cryo-ET is moving from niche to widespread technology in structural biology. Soon we will be able to investigate muscle diseases at molecular and even atomic level. Mouse muscles are very similar to those of humans, yet scientists plan to investigate muscle tissue from biopsies or derived from pluripotent stem cells. Good day, this is Professor Will, CBMD PhD, on microbiology, bioinformatics, and pathophysiology. This video is strictly made for educational purposes only. Well, hope you enjoyed listening. Until next time, have a great day.